moving into our final third of the class, and just to alleviate some fears about Friday, that'll only be a 15-minute long exam, and it's modified so that it will take 50 minutes, whereas your last exam was a 90-minute exam. So this has less material on it, and so then that'll launch us. What we're talking about today is not going to be on that exam, but will be on the other exam. So what you'll be looking at is material that came from Dr. Childers, Dr. Chen, and Dr. Petrovic. So that will be probably a lot like the last exam. There will be some mix of some short answer questions that require some writing that should take less than 10 minutes, and then some other things that are ultra short that would take about 1 minute each. So that's what we're looking at. I think we've moved away from anything that might have math in it, which I think scares some of you. I'm not exactly sure why, but you're not going to need any pocket calculators as we move forward. But I would like to interject a lot of research data so that you can begin to get used to evaluating it and looking at it fairly quickly. So with that said, we're going to move beyond the G protein coupled receptors that Dr. Childers told you about, and now begin to look to see what they actually do. We're going to call this signal transduction. Today we're going to be looking at what we call second messengers, and the first one that we're looking at today is going to be cyclic AMP. So cyclic AMP then signals its response through regulated kinase. So what we're going to talk about today is how that receptor works. Now, you pretty well were inundated with G protein coupled receptor lore for the last week. But in addition, in the next 3 weeks or so, we're going to also be talking about receptor tyrosine kinases. And what you saw from the G protein coupled receptors had 7 transmembrane spanning helices as its structural conformation. So virtually all of those proteins are going to have this same kind of conformation. And it's one of the largest signaling receptor family that we know of. And in fact, better than 60% of all of our drugs are based on cell signaling that goes through this particular pathway, which is why we're putting so much emphasis on it. But another pathway that's very important for signaling, and particularly cell cycle, apoptosis, pathways, neuroprotection, all go through what we call receptor tyrosine kinases. And these span the plasma membrane twice. They're dimers, actually, so each one spans once. For most of these, the ligand is going to be binding to the outside and will contribute to allowing dimerization. The only exception to that is the insulin receptor, which is already dimerized, and those dimers are held together by disulfide bonds. Once these are dimerized, then the thing that makes them called a tyrosine kinase is that the inside of these receptors now can phosphorylate the tyrosines that are on the opposite partner. And then you can use this as a scaffold. Many other proteins can bind to it, and that sets up the signaling pathways through those. A very similar pathway is called cytokine pathways. And here you can see the same dimer format, but in this case, the tyrosine kinases that are on here have to come in from the cytosol. They're not already built into the receptor. So we'll talk a little bit about some of those. Some of the things we're not going to have so much time to look at are TGF-beta receptors. These work through signaling by phosphorylating some transcription factors that can go directly into the nucleus. We'll just spend a little bit of time on those. But hedgehog receptors, Wnt receptors, and Notch receptors, we're just not going to have so much time to be able to work on those. But you can see that they can interfere again, the goal for these is to go right into the nucleus and regulate that way. Now, just a review of what Dr. Childers told you, the signaling through these 7 transmembrane receptors goes through a variety of G proteins, and they're all named based on what kind of function they have. So the one that's named GS stimulates adenylyl cyclase. That's an effector molecule for that G protein. It takes the G protein that one step further so that it can take ATP as a substrate and make cyclic AMP. So now we call the cyclic AMP the second messenger, because it's the cyclic AMP that can go into the cell and regulate effects. And it does it through regulating one primary kinase called protein kinase A. The A comes from the A in cyclic AMP. You could also say the A could come from the A in adenylyl cyclase. So the S on the GS is because it stimulates adenylyl cyclase. But there's a family of G proteins called GI, which inhibit adenylyl cyclase. So it makes the cyclic AMP go down in the cell. This is a multiple family. It's got 3 GIs, and those are different gene products. It's got GO in that family, and GO has 2 splice variants, A and B. Then we've got some homologous proteins to the GS, and one of them is found in the brain. Particularly, it was initially found in the olfactory system, so it's called OLF. But it also works with adenylyl cyclase, and it stimulates. So it's really quite homologous to the GS. Then we have some that are not so homologous. The GQ does not work through the adenylyl cyclase like these others do, but it works through phospholipase C. And we'll talk about that on Wednesday. We also have a cyclic GMP phosphodiesterase, and I'm going to talk to you about at the end of today's lecture, because this is only found in rod outer segments and cones, and the receptor for it is rhodopsin. This is a very concise set of pathways, and the effector being a phosphodiesterase, you can see, is very different from either the adenylyl cyclase or the phospholipase C that we have in the rest of the body. But we're going to go over it, even though it's restricted to the way visual system works. It is extremely important, because if there's a glitch in any possible one of those proteins, it can lead to retinitis pigmentosa, other kinds of diseases causing blindness. But more importantly, to our general phenomenon in signal transduction, almost everything we know about how the G protein coupled receptors work, turns out we've learned from the people that are studying rhodopsin and its signaling in the rod outer segments. So we're going to just go over that a little bit. So that's kind of a summary of what we're going to take a look at today. So we're calling this second messenger systems because the receptors themselves are on the plasma membrane of the cell. And at that place, they can recognize hormones coming from the bloodstream, or cytokines, or other interacting proteins that may modify the reaction. And they're also going to be having some internalization, which we'll talk about a little bit. But essentially, the response to the hormone is occurring at the outside of the cell. So the transmission of the signal has to come from something that can be occurring on the inside of the cell, and that's going to be the ability of the adenylyl cyclase, in this case, to make cyclic AMP. In Wednesday's lecture, we'll go over a homologous protein called cyclic GMP, and you'll see that there's some similarities between the adenylyl cyclase and the guanylyl cyclase, but the signaling is really quite different. But you can see that the only difference here is that this is an adenosine and this is a guanosine. But it makes a lot of difference inside the cell, because this is the trigger for a kinase that can phosphorylate substrates called protein kinase A. cyclic GMP is a trigger for protein kinase G, and that works through very different kinds of substrates and recognition. Now, the second messengers that we're going to talk about on Wednesday are those that come from the phospholipase C. One of those is diacylglycerol, and that was derived originally from the phospholipids that are in the plasma membrane after the head group has been removed. So you'll see what we have is two fatty acid chains. That's what this diacyl is all about. And the backbone is the glycerol. So this is diacylglycerol. And it can activate another protein called protein kinase C. Then what's released is that head group that used to be down here. And that's a diffusible product because now look at all these phosphates. That makes them water soluble. So they can leave the plasma membrane and they can open channels in the endoplasmic reticulum. So these are also second messengers. So these are the second messengers that are important for signaling from these G protein coupled receptors. We're going to limit ourselves to talking about cyclic AMP today. But you'll see one of the things that they all have in common, including this one finally, is that once they have gotten into the cell, they have a specific kind of kinase that they interact with. And the kinase then can promote the response by phosphorylating specific substrates that are specific to that cell. So you can see how one receptor, depending on what cell type it is, can produce multiple responses. And you can see that if you have multiple cyclic AMPs produced, that you can stimulate and trigger a lot of enzyme activity just from interacting with one or two receptors on the plasma membrane surface. Okay, now our diversity here in our G protein coupled receptors can be broken down into multiple different pathways and families of G protein coupled receptors. So did Dr. Childers go over this with you, the different kinds of pathways and families? No? Okay. Well, so the first G protein coupled receptor that was actually discovered was the rhodopsin. And it is going to be in that rod outer segment for the ability to trigger a neuronal response from light. And this now has created a very large family of homologous G protein coupled receptors that have great similarity, and including many that you look at here that are going to be responding to neurotransmitters, but also some that are responding to odorants. So while the retinal and the rhodopsin are triggered in the rod outer segments, the odorants are triggered in the nasal passages and send their messages in to work on several areas of the brain that can respond to the different odors in the environment. You'll see down here that we have another similar family that responds to peptides, cytokines, and some of those are going to be triggered by working through a protease that can break off part of this receptor here on the N-terminal and then use that new N-terminal as what would be the ligand that would bind to the receptor. So now it can take this after the protease has cleaved off much of this N-terminal region on the outside of the cell. It can use that as its very own ligand. We have some that are going to be going through very large N-terminals on the outside of the cell that are going to be responding to some very large hormones like LH, TSH, and FSH that are about 60,000 molecular weight. So you can see that at this point we have a much larger ligand than some of these small ligands that tend to be binding and nestled inside where the G protein coupled receptors are located. Then we have family 2. This was a family that is largely gut hormone receptors, and these are binding, since they're all N-terminal rather than inside the cell. And then our third family are ligands that are actually very small, like the amino acid ligands. So in bacteria, this family is largely there to identify amino acids that are going to be in the environment so that they can use them and indicate that this is maybe where some nutrients are located. But this has evolved into our very important neurotransmitter receptors, the metabotropic glutamate and metabotropic GABA receptors. So you can see they've got the same transmembrane region, but they've got what is now a very, very large N-terminal, and it's got the ability to close over its ligands. And so this is called a venous flytrap configuration or structural motif. And you can see that in addition to small amino acids, this also is triggered by calcium. Calcium, of course, is very small as well. So you would think it doesn't sound right to have such a large N-terminal for such small little ligands, but this makes this greater complexity here. And most of these receptors have a lot of allotropic and influences from other kinds of triggers in the cell. So this is a family unto itself. And you can see how some of these are related to each other. Most of what we are encountering with our hormones and our neurotransmitters are probably up here in family 1. Okay, so family 1 is how we're going to be interacting with adenylyl cyclase. And this is our 7-transmembrane receptor. Our agonist diffuses prototypically to find its way either to ligand with the N-terminal up here outside the receptor, or inside where these 7 helices form a kind of a barrel. So once that happens, the general prototypic response is that GS is triggered, and it dissociates so that the alpha subunit is dissociated from the beta gamma subunit after the receptor has triggered the removal of the GDP and the GTP comes on. So once that happens, this G-alpha S protein now can trigger the adenylyl cyclase. It uses ATP from the cytosol as a substrate and makes cyclic AMP. Yes?
For adenocyclase activation, is it always the GAS subunit, or could it be sometimes the GB?
It could be the GOLF, but that's really limited pretty much to the brain. So the stimulation is going to be from the GS family.
Okay.
Okay? Then prototypically, once our cyclic AMP is in the cell, it triggers a kinase, the protein kinase A. Now let me just explain to you a little bit about how the protein kinase is configured and how it recognizes this small molecule. This is not drawn to scale, by the way. So the protein kinase A has 4 subunits. 2 are regulatory and 2 are catalytic. So catalytic means that these are the subunits that actually perform the phosphorylation of substrates that are in the cell. So these 4 proteins are bound generally to each other in the inactive state. And when cyclic AMP triggers them, it's got 2 binding sites on each one of the regulatory subunits. When each regulatory subunit gets bound, it releases these catalytic subunits from where they were previously bound to the regulatory subunits. And now these catalytic subunits are free to diffuse into the cell where they can encounter some of their substrates. Now you can see we've got this little bar over here called AKAP. We pronounce that ACAP. It stands for A kinase anchoring protein. So A kinase anchoring proteins are generally going to hold these protein kinase A in the vicinity of where the adenylyl cyclases are. So this cyclic AMP does not have to diffuse widely within a cell to look around and try to find its protein kinase A. It will probably be very, very close. So when we talk about diffusion, we can keep our cyclic AMP fairly close simply by having a scaffolding protein like the ACAPs. So many ACAPs are now being found to actually bind to an adenylyl cyclase or find a way to be close to an adenylyl cyclase. So that when this whole program happens, it happens because these proteins are all in close proximity to each other. So you could pretty much envision that they must be in a kind of a complex that is not simply proteins diffusing around in the cell membrane and waiting to find each other. Okay, now the GI proteins, on the other hand, are inhibitory. And you can see this is the schema we just looked at. Some typical stimulatory hormones include epinephrine through the beta-adrenergic receptors, glucagon, adrenocortical stimulating hormone, ACTH, that's a peptide. So these all have a site on the adenylyl cyclase. The GI proteins also have a site on the adenylyl cyclase. So they respond to such hormones as prostaglandins, some of the adenosines. My favorite hormones, the cannabinoids, are going to be binding to their receptors, working here through the GIs. Then the GI is released in the very same way that the GS is, so that the alpha subunit can find its site, and this will reduce cyclic AMP levels. So if you've got both of these working together, then this is going to modulate the ability of the GS-stimulated receptor to actually have an effect. Now, I really do like looking at structures, and for those of you who like to see structures, I've got lots of them in here for us. I guess that's good. So this really can help us visualize a little bit about what we're looking at here, because this was your well, maybe you can tell me, what is this?
It's the G alpha.
It's the G alpha subunit. And so it's binding its little GDP right in here. It's got, what is this half of it called?
The GDP binding.
The GDP binding site, yes. And over here, this other half is called the helical domain because it has some helices in it. So once this is triggered by the receptor is going to release this GDP. The cell has a lot of GTP already in it. A GTP is going to bind to this very same site that's going to activate it. So we've got what we call some switches here. This little site here is a switch. This little site here is a switch. And once the GTP gets bound, it's got one more phosphate on it. It's a little bit longer. It triggers these switches now so that it changes the conformation. And you can see we're no longer bound to the receptor. We've dissociated. Now we've got this alpha subunit that's free. And once these switches are activated by having GTP on here, we now have some sites that are going to be activating the adenylyl cyclase. And since this is no longer associated with the receptor, we've got this whole surface area now to be able to interact with the adenylyl cyclase. Now this is a schema of our adenylyl cyclase. It's got 6 helices. In this case, it's different from the G protein coupled receptors that have their N-terminal outside. This one has the N-terminal inside. And then it's got this little part that is inside the cell. And this is where the work actually happens from this protein. This is our catalytic domain here. Then we have what looks like gene duplication. When you see a part of a molecule that looks exactly like the first half of the molecule, you got to figure that probably somewhere in evolution this was duplicated. And sure enough, this looks exactly like this. So we've got another 6 helices, and then we've got the C terminal over here. And the C-terminal is also important for catalytic activity. So let me show you a little bit about that. So for those who like the gene expression format, we have our N-terminal, we've got our sites that are going to be going through the membrane, called the membrane sites. Then we've got our catalytic site. Then we've got another set of membrane sites, and then we've got the other catalytic site over here, and the C-terminal. But what it really looks like when it's inside the membrane is that all of these helices have hydrophobic residues outside, so that's how they get themselves bound into the membrane. And you can see what's happening here is that this little catalytic domain is contributed by this, and this catalytic domain is contributed by this, and they're held together in the inside of the cell. So when the X-ray crystal structure was done, getting transmembrane proteins out of the membrane is really difficult. You have to use a lot of detergent. And then you can't get good crystals to find the structure. So a very brilliant postdoc in Al Gilman's lab figured that what we could do is get rid of all of these and make a recombinant protein that only has this and this. And that's exactly what they did, and this is what they ended up with, was the two catalytic domains. One is shown in gray, the other one is shown in blue here, and these are now interacting with each other. And that's how they could get the crystal structure to be able to tell us how this was working. And very, very lucky for us, those sites for GS alpha and GI alpha happen to be contained right here. So we really don't need these greasy helices structures for the adenylyl cyclase. We could just use this. Now you can see that what's inside here is going to be our ATP binding site, because that's going to be the substrate. And the site that is going to be on one side of this catalytic subunit actually takes the ATP. But then when it's hydrolyzed by water, the cyclic AMP and the remaining pyrophosphate are delivered to the other half of the catalytic site. So that's why you need both of them. You can't function with only one of these. You have to have one that takes the substrate and the other one that accepts the product in this particular enzyme situation. Now, what would you, if you were thinking about, if you were God and you were going to make proteins, what would you do with something that has 12 transmembrane helices? Why would that make a nice kind of a protein to make? They could anchor something, yes. What could they do functionally after they've got something anchored? Transfer something, yes. So this could be, this could be a transporter. And sure enough, this kind of motif is a transporter for glucose. So, you know, the family of evolution here is, you take a glucose transporter, you want to use it for something else, you somehow get some gene duplication, maybe it has a little enzyme on it, maybe it's an enzyme that looked a lot like guanylyl cyclase. You put the whole thing together by a few of these mutations, and you've got a brand new enzyme. So that's probably what happened somewhere along in evolution. And so this structural motif here looks a lot like glucose transporters. But the adenylyl cyclase doesn't have any transport activity. It just has these catalytic subunits. Now, one thing that we've been able to discover, and this was from folklore medication, was there was a plant product called Coleus forscoli. So do you all know what a coleus plant is? You probably have somebody that you know who has one sitting in their windowsill in their kitchen. It's that plant that they put out in the summer that has all the bright red leaves, And unfortunately, for those of us that like to plant things, it doesn't survive in the winter, so we have to replant them. But that's the coleus plant. And in folklore medication, it was used for people who had congestive heart failure because it had some product in it, which we now call forskolin, which would trigger the beta-adrenergic receptor kind of response. So it would act a lot like epinephrine, so that it could increase the force of, increase the rate of contraction of the heart. So it doesn't do it by activating the beta-adrenergic receptor. What it does is skip the receptor part altogether, skip the G proteins, and go directly to the adenylyl cyclase. And the way it works is this is a very lipid-soluble compound, but it can bind right there where these two catalytic components are. So it can hold them together. And simply by holding them together, the likelihood of an ATP slipping in is greatly increased, and you can actually stimulate a lot of cyclic AMP production from having this little tiny molecule that can hold these two components together. So when you're reading the research literature, you're going to see a lot of papers in which the researchers said that they gave forskolin, and therefore they made cyclic AMP go up. And if you don't have a good appreciation of how this works, you could get yourself very confused, because you might think, oh, it might be having to work through a G protein coupled receptor. Well, it doesn't. It gets into cells because it's lipid soluble. It can pass right across the membranes. It can interact. It can make these catalytic subunits come together and force them to work. And you can get quite a bit of cyclic AMP made in the cell using forskolin. So you'll see that a lot in the research literature. Okay, so now we've got our G protein. We had our helical side, right? We have our side that binds the GTP. This is how it interacts with the adenylyl cyclase. So this is those two catalytic units. This is forskolin, right in the middle, holding them together. But you can see that it can still bind those switches that were on this side of the G protein. So even though it's got forskolin in it, it can be boosted even further if you have a GS. So the GS can bind, the forskolin can bind, and you can get a lot more activation than you had before. So here's that very same kind of structure. Now it's really embedded underneath these two sets of transmembrane sites. Our little forskolin is here, our ATP binding site is here, our GS alpha is over on this side. And then you can see that we can have our GI alpha over on the other side. So what we've done is switch this around. Right now you can't see the beige part very well, but over here you can see one catalytic site on the top, one on the bottom. The GS is interacting with one of them. The GI is interacting with the other. And when you look at what's happening down here in the middle, you can see that this ATP is binding, so that when ATP binds to the substrate on the purple catalytic part, once it's hydrolyzed and the phosphate comes off, now the phosphate is going to be binding over here on the other side, the brown side, and the cyclic AMP is going to be released. Now, just to make sure that you're not confused, I'm going to confuse you even more by letting you know that this prototype that we've just looked at, that you're all going to go home and think you got to memorize, is not the only way that we can regulate these. And so if you think this broad generalization of cyclic AMP goes up, the cell gets activated, is going to be true for all of these, let me tell you that we have 9 different kinds of adenylyl cyclases. We've got 3 that are lumped together, 1, 3, and 8. So they were named based on the when they were discovered, starting numbering with 1. So the ones that the Gilman lab looked at and crystallized were actually number 1. They're actually stimulated by GS and inhibited by GI, but they have a very important region in which they can trigger a response to calcium. And they do this because they've got a binding site for a very small protein called calmodulin, which is a calcium sensor inside the cell. It binds to calcium, and it's got a binding site that is on this family of adenylyl cyclases, so it stimulates here. So this is really very important in places where calcium regulation is important. And that could be in neuronal cells, it could be in cells that are responding to the GQ-coupled receptors that trigger a calcium mobilization inside the cell. Then we have the ones that are called 2, 4, and 7. Let me skip over those for a minute and go down to types 5 and 6. So type 5 and 6 were the ones that we just talked about as prototypes. They are predominantly regulated by either GS or GI. And this isn't right. Take the word stimulate out. I thought that I had taken it out, and evidently it didn't get saved. So the GI inhibits, the GS stimulates. So that's what you're seeing over here. GS stimulates, you've got a little positive sign. GI inhibits, you've got a little negative sign. But pay attention now. This is not the only regulation, because this can be phosphorylated by the kinases, PKA itself, or the other kinase, kinase PKC, and in this case, C stands for calcium. So we've got two different kinases. And then our lab actually discovered that nitric oxide can negatively regulate. It can inhibit as well. Now, why might you want to have PKA inhibiting this adenylyl cyclase? If you were trying to organize a great schema of how the cell is going to work. Some reason that you might want to have this?
You have high amounts of PKA. You don't need the adenylyl cyclase.
Yes! If only you could speak louder so that the people on the other end of the room could hear. So if you've already made a lot of cyclic AMP and you've already triggered the protein kinase A to go out and phosphorylate things, you don't need to make cyclic AMP anymore. So if somebody came to your house and they were ringing your doorbell all day long, believe me, after the first couple of seconds or minutes, you're not going to pay any attention to that anymore. But if they stop ringing the doorbell and then come back an hour later and ring, then you're going to come back and answer the door. So same deal here. If you've got a lot of cyclic AMP, you've got to stop making it at some point, or your cell's not going to respond again. So once the protein kinase A has been triggered, it feeds back to shut down the adenylyl cyclase. Now this other one, the 2, 4, and 7 subtypes, these have a very perturbing way of doing things, because now they're stimulated by GS and they're inhibited by GI, just like the others are, except that once the beta gamma has been released, it augments that stimulation by GS. So you can actually release something from a GI-coupled receptor, and the beta gamma is going to have the opposite effect of the alpha, and it is going to now augment this. We call this a synergism, so that it actually works a whole lot better, and you'll get much more cyclic AMP made when you trigger one of those GI-coupled receptors. And then we've got the same deal here. The PKA inhibits it. PKC can either inhibit somewhere along this pathway or can stimulate. So you can see we've got lots of ability to regulate. Now I brought you some real data, and I've got 4 different cell types here. So I'm going to pass this around so that you can, if you don't mind getting up and stretching for a minute. Anybody who's interested in macrophages, come on over here to this side of the room, and you can talk about number A. People who are interested in glutamatergic neurons, get up and come over to this side of the room. Melissa, you're interested in glutamatergic neurons. People who are interested
steal all of them.
If there are more than 5 of you in a group, whoever's the last one. Yeah.
Oh, perfect, that's what I wanted.
Okay, so what I want you to do, there are
We wanted to
four
do.
different bar graphs there,
It's not
A,
enough
B, C,
for
or
everyone.
D. With your group, talk about what your interpretation of that bar graph is, and you've got about 3 minutes to do that. And elect one of the people in your group to explain what you've discovered. So this is the figure legend. It's got the methods
Sure.
in it. And I'll turn on the lights so that you have something to think about here. And I'm going to leave this figure on here.
There's a little slide
This figure here is something you'll probably want to look at a lot in order to
Crazy.
interpret your data.
She's really into, yeah.
It'll start another video, but it always takes forever for it to like ligate together. So don't say this for Oh, I don't know what any of these words mean. Hold on, I know that one.
Well, heaps is just a buffer.
Oh yeah.
And the medium is just the medium that you grow cells in. What is that?
ACL2?
Oh, let you. What is what? ACL.
Oh hey, this helps more. Read the bolded stuff. Primary culture as follows: A. Microglia, B. Glutamatergic neurons, C. Medium spiny neurons, and D. astrocytes. Cells with intestinal facilities like A. Reproduction in response to G or WI plus human with agonist compounds that stimulate cannabinoid which are GPCRs. A and B cells were stimulated for forskolin animals or ionomycin calcium ionophore has been present for actors of APC or WI. In C and D, the cells were stimulated by is where indicated.
Or I'm confused. I mean, so pretty much the basal levels. Why would
Do you all know what ionomycin does?
no?
It's an antibiotic that punches little holes in the cell membrane so that calcium can come in. So if you've got calcium in your extracellular medium, which you are if you're a cell and you're going to be bathed with extracellular fluid, you're going to have millimolar concentrations outside. It's going to rush into the cell. So now you're going to get a lot of calcium in the cell.
What does calcium have to do with? I don't know. It decreased the.
Is that how antibiotics work? What is the calcium?
I don't know.
Why does it matter?
The only thing I'm getting
I mean, typically calcium means like contractions or stuff like that.
So ionomycin would probably just.
calcium, adding calcium is one of those. TKA, right? The blood was another word for the blood. Calcium. Not this one.
Are you talking about Petrovic's lecture?
That wrote up with
I remember calcium in Petrovic's lecture. There's a specific.
the guy.
The very
Shoulders
first.
mentioned. Oh no, I'm sorry. The woman, the very last woman mentioned it. She was like,
Dr.
"You don't
Chen?
really have to know this." Yeah, it was that diagram.
She's like, I'm not to know it though. Alright, hold up. A and B was
Ionomycin.
which are agonist complexes.
So ionomycin reduces cyclic AMP accumulation.
What is A? Oh, they're both agonists.
Okay, so diagonal stripes is control. More stripes is iono.
Because how come here's having totally opposite effect? The biggest effect.
And they treat it with ionomycin and FX. And then this one we have is
No, that's fine. I joined it.
oh, A is microglia, B is glutaminergic. Why?
Yeah.
Why were the microglia not treated with HUWIN?
So astrocytes, HUWN, actually increase accumulation.
Well, so clearly there is a significant change with glutamatergic. I think
That is not the case in
glutamatergic.
AG and then in A and B,
Oh, glutamate, not glutamate.
ions plus FS.
Glutamatergic neurons have a much greater
So the ionomycin and the
camp accumulation when treated with ionomycin and
Ionomycin, yeah. And then for
Wait, aren't we only supposed to be doing A?
Are we only doing
wait, what are we doing? What are we
Everyone
doing?
has the same one. We're doing A.
What does that mean? What are we doing?
We're just, I guess, explaining what it says and why.
It's just microglia that have been treated with different agonists.
And then it just tells you cyclic AMP accumulation. So you have the basal level, and then when you add ionomycin, the cyclic AMP,
They're agonists.
It
It
barely changes.
yeah.
It's probably not a significant change.
What it's saying is that, like, if you add either agonist, either HUWN,
Forskolin?
they seem to have about the same effect.
Oh yeah.
Yeah, so like only the agonist doesn't do crap and only the ionomycin doesn't do it. But together they both combination, which makes sense. Just activators only just said the ionomycin, you know. So of course you're gonna get more cyclic to go in. I think that's what it's saying. Does that make sense?
What one is an activator?
Okay, are we all ready to,
Because you need an ionomycin.
So then it makes sense. So it shows that ionomycin has no effect.
Because doesn't the agonist bound to ionomycin?
Yeah, punches holes to allow calcium.
I guess why
I don't know.
It probably just CAMP accumulation and
Okay, well, you can come back to your
Maybe you're
seats
right.
and,
I don't know.
be sure your group has
Because this is
elected
just
the
for
speaker
scaling. You don't
to-
have
I
ionomycin.
think you're right because that's what it's saying, that you don't need ionomycin.
Yeah, it's saying you don't need ionomycin. You don't need the calcium
Because like you said, it's on the
influx.
membrane.
Does
Okay.
You want to explain this?
does anyone who
So what you're saying is that when you add ionomycin by itself, there's no change, right? When you add forskolin, you see a big change. When you add them both together, you still see the same change, which shows you that ionomycin isn't adding anything to the forskolin effect. So it means that whatever ionomycin is doing, which we're told is that it allows the CA influx. It doesn't really help your camp accumulation.
model cells in culture. So, actually, we don't know, very much
You
about
got
this, but they
this.
were grown in a cell culture.
This woman.
They have fresh medium, and why were they given treated adenosine?
You can measure the adenosine. It's radio labeled now, so you can
Yes.
actually measure.
You can measure adenosine, but what do you really want to measure?
Radioactivity.
Cyclic AMP,
Right.
which adenosine will be equal to.
Not exactly. So what is the adenosine used to make first?
ATP?
ATP. ATP is the substrate. So your adenosine is going to be incorporated into ATPs. You'll have a lot of now radio-labeled ATP. So now when the ATP that's already there in the cell that you just made, and let me see if it says what you did, that was like overnight. and then you washed it away. So now the only radioactivity you have in there is going to be what the cells have already made into ATP. So now, when you stimulate this response, now you get radioactive cyclic AMP, and you can separate those. So, so we also have some HEPIs. Why do we have HEPIs in there?
Ion balance?
Yes, it's ion balance. It's a buffer. It's going to keep it at pH 7.2 or 7.4. We've got some fatty acid-free bovine serum albumin. Why do you suppose we might want to put that in there? Well, I think it's one good way of carrying in some of these lipid-soluble things like ionomycin and forskolin. Forskolin is lipid-soluble. So what did you learn from your pharmacokinetics about how lipid-soluble compounds are transported in the blood? They're carried on what?
Proteins.
Not cholesterol.
Proteins.
Albumin.
Yes,
So you're mimicking this. So you're carrying in things like your forskolin. Okay. Phosphodiesterase. I'll tell you ahead of time, phosphodiesterase breaks down cyclic AMP. So if you're measuring how much cyclic AMP is made, you don't want it to be broken down. So you put in one of these, an inhibitor from Roche. You remember I told you that before a drug gets to be marketable, you use the code name that the company gave it. So this is RO from Roche, 20-1724. And you can buy it in catalogs that way. The other phosphodiesterase inhibitor, 3-isobutyl-1-methylxanthine, what does that resemble chemically, do you suppose? Have you heard of methylxanthines as a class of drugs? No? Caffeine and theophylline. So theophylin's largely in tea and caffeine is largely in coffee. And these drugs are all methylxanthines. And it turns out that they inhibited these phosphodiesterases. Okay, now, you know, we can't get away without talking about cannabinoids because they're my favorites, but this could have been any GI coupled receptor, and these are put in this assay. And the other thing is the TSH. So what is TSH? Okay, thyroid stimulating hormone, and that's a GS coupled receptor? Maybe? Okay, so with that kind of background, what they did was treat the cells with all of these things for a certain period of time, and they were at 37 degrees, right? You're at 37 degrees, so if you function, cell culture probably functions at 37 as well. And then they removed them, and they hit the cells with perchloric acid. Now, why would you do that?
Open them up.
It blocks the activity, yes. In fact, it's probably pretty nasty. You've got 2.5% perchloric acid, so you probably not only have blocked activities, but now you probably denatured a lot of your proteins. So if you've denatured your proteins, they're not going to continue to either make or break down cyclic AMP or respond to hormones. Now you've just got this mess. And so you neutralize it with KOH so you can work with it. And then it's got this two-step column separation. And they've got a reference to Solomon in 1991. So this is a kind of a standard old technique that separates your tritiated cyclic AMP from your tritiated ATP, which your cells had already made. So now you can measure how much cyclic AMP. So those are the general. So now, how about our microglia people? Who's microglia? Okay. So tell us about figure A.
So in figure A, we're looking at microglia cells and they're...
Why don't you stand up and shout so that the guys in the
I
back...
won't be much taller.
I only have you by an inch.
Alright, so in figure A, we have microglia cells looking at their cyclic AMP accumulation. So first we look at, from left to right, the first bar on the left is the control. First we compare it to the next bar, which is treated with ionomycin, and we see there's really no great significant change in the cyclic AMP accumulation. And we compare that control to the third bar, which is treated with forskolin, and we see that there's a very significant increase in cyclic AMP accumulation. So we can kind of infer that forskolin is having a greater effect on that cyclic AMP. And then we look at the last bar, the bar furthest to the right, and it is a combination of ionomycin and forskolin treatment. And we see that it has no really great difference from the bar that's only the forskolin treatment. So this kind of implies that the ionomycin is not really having any effects on cyclic AMP accumulation, and it's really just the forskolin.
Okay, so what can you tell us about this schema over here, about what must be in these cells? Do they have, would you suspect that they have 1, 3, and 8 adenylyl cyclase, which responds to calcium?
No.
No. Okay, good point. So now we've got a choice. We probably have either this family or this family. And right now, we haven't got any other hormones in there, so we can't make any inferences, right? Okay, so let's see if the people in B, which is our glutamatergic neurons, so that's in the back half of the room. Who wants to tell us about B?
Yes.
Oh, yes. Write your notes on it. You can write your notes on this. I'm not collecting them up. So what's going on in B?
In B, the basal state, there's got basal rate of cyclic AMP production. When you add the ionomycin or the forskolin, you have increased accumulation of cyclic AMP. However, when you combine the two together, there's significant increase, and you add in the HU or the WIN, which is like an agonist. There's not much. There's increase, but not as much when you just have the ionomycin and the forskolin alone.
Okay. So what can you infer about what's going on in here? You don't know? Who else is in the group? Help him out. Okay. Well, the WIN and the HU are binding to a cannabinoid receptor. What kind of a G protein do you think that cannabinoid receptor must be interacting with?
Maybe inhibitory?
Inhibitory. Yes, because, as you said, the cyclic AMP levels were decreased compared to what happened when you had forskolin and ionomycin by itself, right? So that decrease would be typical of a GI interaction, right? Okay. So what can you tell us about which ones of these this probably is? Which adenylyl cyclase family might be working with in this particular cell? What was your main stimulator in the first 2 bars, the first 3 bars? Well, we can make it the first 2 bars. Calcium coming inside stimulated. And then you saw when calcium and forskolin were together, you had a huge synergistic response, right? So what does that tell you about what kind of adenylyl cyclase this must have been?
2, 4, 3, 8.
Why 2, 4, and 7? Somebody up here that wasn't part of your group said that. Why would it be 2, 4, and 7?
Because they have a synergistic effect that results in most of the
Well, what synergism was it? What is being synergized?
beta gamma has
Okay.
beta gamma has an
But-
opposite effect of
But
alpha.
we already said that we were seeing inhibition. by this GI. So that doesn't sound like a synergistic increase, does it? So I think we could eliminate, actually, this group here, because we saw an inhibition, we didn't see a stimulation. But what was synergizing?
This is the forskolin
Yes?
with the calcium.
Yes. Which one of these responds to calcium with a big increase?
The first one.
1, 3, and 8. So what kind of adenyl cyclase does this cell have?
Calcium chromoglobin.
Mm-hmm. Yes.
GS.
Yes.
GI.
Yes. Okay. We didn't actually have any hormones that stimulated here. But we had calcium. So you've got to know that it's got to be one of the top family, right? It responded to calcium. Okay. Where's group C? C are the ones that we said were medium spiny neurons that's in the back here, right? So tell us about C. C is easy.
Okay, so we have the cannabinoid receptors that are located on the median spiny neurons. And so with no cannabinoids, we see this high level of cyclic AMP accumulation. And then when we use two different agonists to bind to those cannabinoid receptors, we see this significant decrease in both of them.
Okay. So what was this being stimulated by?
Forskolin.
Okay. Was it stimulated by TSH?
No.
Okay. So we don't have that on there, but it said that there was TSH stimulating someplace, right?
Yeah. Or it says FS or TSH, but in this one it says
Yes.
just Forskolin.
Right. Okay. So if it's or, and we could have seen that response even though they didn't have it in their bar graph here, what would that tell you about this response?
That it's an inhibitor. It's acting by probably GI.
Okay, the inhibition.
The inhibition.
And this was a pretty big inhibition, right?
Yeah.
Okay. And it could be stimulated by TSH, a hormone?
Yeah.
Okay, so what would that be? Which cyclase over here gets most of its stimulation from a hormone and that goes through GS? The bottom. Okay, so type 5 and 6, they're not stimulated by anamycin. And actually they didn't show us that here, but we knew that, even though they didn't have it in there. So all we see is that we've got a stimulator and we've got an inhibitor. Okay, what about D?
All right, so for D, we kind of went by process of elimination from bottom up on this one. So we added, or the experimenters added the ionomycin, so that's going to increase calcium cellularly, and then forskolin's present in all three bars. And then the last two darker bars have the two cannabinoid agonists. So we, from control of just forskolin and the calcium ionomycin stuff, we see an increase. So going up from the bottom, it's not type 9. We added forskolin and we have a response, so eliminates that. We said it was not type 5 or 6 because calcium did not have an inhibitory effect, so eliminated that one. And then we kind of jumped up and said it might be type 1, 3, or 8 because calcium's present, and then when an agonist stimulates, we have an increased response, so we thought it was 1, 3, or 8.
Okay, so, but is calcium there?
Isn't the ionomycin bringing in or allowing calcium to enter the cell? I thought that was one of it. Might be very wrong on that.
Okay.
Yeah.
Yeah. In
Okay.
C and D, the ionomycin wasn't added.
All right,
Look
we'll
that
put
up.
that.
Okay, so let's assume there's no calcium going in here. So what happens when you add these two GI coupled receptor hormones now, in contrast to what happened in C?
Well, if there's no calcium in it, then it's gotta be 2, 4, or 7, because that one doesn't have any calcium interaction.
Right,
And we already eliminated 5 and 6 and 9.
right. So now you've got a beta gamma that's being released that's triggering this augmentation.
So that first white bar, I thought, if we look at the figure A, I thought that was ion plus FS. C and D are totally separate.
Yeah, this is, yeah. Okay, the next time I doctor this up, I'm gonna move it so that A and B are not close to C and D and renumber them.
So the
So
the
this,
coloring
this,
legend.
right
for-
here, this legend is.
Oh, I see.
is
I
over here.
missed that. Okay.
Okay.
No, that's fine.
Alrighty, so you can see same kinds of deal here, but 4 different kinds of cells that have different kinds of responses. So you've got something that you can go home and memorize, but on the other hand, keeping in mind that, that you've got 9 different kinds of adenylyl cyclase, you may see some different things that you're going to have to interpret based on the kind of adenylyl cyclase in your cells and other things as well. So our cyclic AMP levels are going to be modulating not only through the kinases that are PKA, but here we have another small group of protein called EPAX. And this is an abbreviation that tells us about guanine nucleotide exchange factors that are going to be helping some of the small G proteins to make a response by exchanging their cyclic AMP or their GDP and allowing GTP to bind. So this is a small family that works with small G proteins. And did Dr. Childers talk to you about small G proteins? These are like Rho, RAC, RAS, RAF. No? Okay. So I'll tell you a little bit about them maybe a little bit later on. But these are small G because instead of having the 40,000 molecular weight alpha subunit that has the helical part on it, that part is missing. You only have the GTP. This whole series of small G proteins perform lots of functions in the cell. And RAS is one that has been crystallized. We know a lot about it because it was an oncogene. And it regulates binding that initiates the MAP kinase pathway. And we'll talk about that on Wednesday. Okay, and then you see one of these A caps. This A cap is binding a phosphodiesterase, and that's going to be breaking down the cyclic AMP. Now let me give you a little rationale about why we want to begin working through a kinase. So this is the response that was discovered from the Nobel Prize that was won from the 1960s when cyclic AMP was identified to be a second messenger, and it was all related to the ability of the liver to take glucose, make it into glycogen, and then break down glycogen back to glucose 1-phosphate to be released after you've taken the phosphate off so that the bloodstream can get more glucose. So this is a highly regulated pathway, and you can see that this protein kinase A starts the whole thing. So if you had a response to epinephrine, you made a lot of cyclic AMP through the beta-adrenergic receptor, you've got a protein kinase A that can stimulate that glycogen breakdown. And it does it through phosphorylating glycogen protein kinase, which then takes the phosphates, once it gets phosphorylated here, it becomes active, and it can phosphorylate glycogen phosphorylase that actually breaks down the glycogen. So we've got a highly interactive series of 2 kinases, and this whole thing initiates from that protein kinase A, using these as substrates. These are substrates that are in the liver. And at the same time, you don't want your cells making more glycogen. So you have to have a shutdown. So over here, the PKA is stimulating glycogen synthase kinase. So the way we name proteins is you generally take whatever the substrate is and then whatever the cell is doing with that substrate. So a kinase is going to be phosphorylating the substrate is glycogen synthase. Synthase means something was synthesized. It was glycogen. You want to shut this down. So here, when the pKa phosphorylates it, it gets phosphorylated, but now it's inhibited. So your pKa has a coordinated response. It initiates the response that breaks down the glycogen and allows the glucose to be released, and it shuts down the synthesis. Okay, so what we can do is, if we've got decreased cyclic AMP, epinephrine's not around, let's say glucagon is not around, we've just had a nice meal, we ate ice cream, we had lots of sugar, now we're going to want to store. So what we do is, at this point we have a phosphatase. A phosphatase is an enzyme that takes the phosphate groups off. So this is the reverse reaction. So now on this side, we take these groups off, these phosphate groups on the glycogen phosphorylase kinase, and that takes off the phosphate on the glycogen phosphorylase. Now these are inactive. So they won't break down the glycogen anymore. But instead, with all that sugar you've got in your bloodstream, and your insulin is going to be allowing something to happen in the cell, you're going to want to prevent this glycogen breakdown, but at the same time, you're now ready to start storing the glucose as glycogen. So now we have less cyclic AMP. We're not stimulating the phosphorylation. So now we have an abundance of unphosphorylated, non-phosphorylated glycogen synthase. And now that is active, and it can begin putting the phosphates on the glucose so that now we can start making glycogen. So we've got a coordinated set of activities going on in the cell, all coordinated by that pathway. Now it gets to be a little complex, but the other thing in addition to coordinating a pathway is amplification. Because if we had that epinephrine binding, then now we can stimulate several adenylyl cyclases from a single receptor, and we can get lots of cyclic AMP made. Maybe up to micromolar concentrations in a region where we're going to be having our protein kinase A. So now our protein kinase A is activated, and now not only do we have that selectivity that we just talked about from the enzymes that are in those cells, but now we have a greater amount of phosphorylation. So we can amplify here with the amount of the second messenger, and we can amplify here because once those catalytic subunits are released, they can phosphorylate a lot of substrate. So whereas if we were going to be looking at the surface receptors, we might get a response that once it's amplified, the physiological response of glucose being released into the bloodstream in response to epinephrine could be taking place at a much lower concentration of your epinephrine as your ligand. So whereas it might take one of these units, let's say 1 millimolar or 1 micromolar of ligand to get 50% of your receptors activated, you may have amplified your response so that only 10% of your receptors are amplified if you're measuring the amount of glucose that might be released into the bloodstream. So specificity, coordination, amplification are all the reasons that we want to work through a second messenger pathway. Now, if we want to get another set of substrates with our pKa, we can now let our catalytic subunits get into the nucleus, and we can cause changes in gene expression. And we do that by interacting with a protein called cyclic AMP response element binding protein. And most people use the abbreviation CREB. And when CREB gets phosphorylated by one of these catalytic subunits, the phosphorylated CREB is now able to interact with the cyclic AMP response element. So this is cyclic AMP response element binding protein, cyclic AMP response element. And it doesn't do it by itself. In order for it to get polymerase II, which is a fairly large complex of proteins, to get activated, we need to have this CREB binding protein, which is 300 molecular weight. So the thing is, when a yeast biochemist or someone has recognized that they have something that can have an effect and they don't know what that protein is, they run it on an SDS page gel. It comes out at a certain molecular weight, and very often they just simply call it the protein that was at 300,000 molecular weight. So at some point, this protein was determined to actually work with the Krebs in order to facilitate this transcriptional activation, and so it was called Krebs binding protein. So you may see it either way. When you see two different names and there's a slash in between, that means that there were two different laboratories naming something two different ways, and we're still using the nomenclature of one or the other. Okay, so this is how we can use this whole pathway to now stimulate new gene expression. Now, whenever you have something that has been produced to cause a second messenger or any other generalization of phosphorylation, you have to have a way of turning it off. So we could take the phosphate groups off with a phosphatase, but we want to get rid of our cyclic AMP. So we have not just one, but again, a whole family of, here's 10 different phosphodiesterase. And they share some of their substrates because they're not all going to be working with cyclic AMP. Some of them can work with both cyclic AMP and that other little second messenger, cyclic GMP. So one of the first ones that was discovered was named phosphodiesterase 1. It could use either one of those and it'll break them down. And it gets regulated by the kinases PKA, regulated by cyclic AMP, or PKC, C stands for calcium, regulated by calcium. And its hallmark that makes it different from all of these others is that it's got a calmodulin binding site that can allow it to be sensitive to calcium. So what does that tell you as what the trigger for this activation is going to be? Yes, you could use ionomycin or anything else that increases calcium in the cell. So what could increase calcium in a cell? An ion channel. So if a calcium channel opens, you get a lot of calcium coming into the cell, then that calcium is going to bind to the calmodulin. The calmodulin is going to be on the phosphodiesterase. So not only do we have an adenylyl cyclase that can respond to calmodulin, we have a phosphodiesterase that can respond to calmodulin. And in fact, calmodulin has its own family of kinases called calmodulin kinases. And there's calmodulin kinase 2, calmodulin kinase 1, CAM kinases. So these are going to have a binding site for calmodulin as well. So you might want to start thinking of calmodulin as a little sensor that's in the cell, a small little protein. Its job is only to bind the calcium, but then it can interact with all of these other signaling proteins that need to have that calcium sensing regulation. So there's a site on this that binds to calmodulin. Now, for the sense of people who are interested in what's going to be useful as drugs, these drugs have been made, and you can see here this is one of our inhibitors, that isobutylmethylxanthine that we just looked at on this example paper. So here we were seeing it as what would be a useful inhibitor in a cell culture experiment. But actually the isobutylmethylxanthines are regulating the cyclic AMP levels in drugs as well, because they can alter the amount of cyclic AMP that would be in the smooth muscle in the bronchioles. And if you have an inhaler, and the inhaler has theophylline in it, what is it going to do? If you've got asthma, it's going to bronchodilate. And it's not doing it because your inhaler has epinephrine in it. It's doing it because you've got isobutylmethylxanthine or theophylline in there, and that is going to raise the level of cyclic AMP that your own body's epinephrine may have already raised. So this is a very good drug. Clinically, it's used in dementia, memory loss, some of these drugs like nicardipine. PDE2 has a regulation in that it can be stimulated by cyclic GMP, even though it can use either cyclic AMP or cyclic GMP as a substrate. And it's used for some cell types. PDEB is only going to be recognizing cyclic AMP as a substrate. And it's inhibited by cyclic GMP as a regulator. PDE4 is a very popular cyclic AMP as its only substrate. It can get stimulated by PKA. And we just saw that on this. So it's binding here to an ACAP. The ACAP we know already binds to the PKA, and it can get stimulated by that PKA that would be binding here. So why would we want to have, a PKA stimulating a phosphodiesterase? I think we already discussed this. If you've already got a lot of cyclic AMP made, it'll activate your phosphodiesterase. So now you've got an activated phosphodiesterase. It can get rid of a lot more cyclic AMP and reduce the cyclic AMP levels. So it makes it a very coordinated stimulation of the phosphodiesterase. And it can take place as soon as that protein kinase A gets activated. Okay, it's got some inhibitors. There's our RO201724 that you saw in your example. And this other one is a Roche compound called Rolopram. And this also is used for treatment in asthma. Here we've got PDE5. This is only going to be breaking down cyclic GMP. And this is a very, very popular phosphodiesterase. It can be activated by PKA or by the PKG. And its inhibitors are sildenafil and Zapronast. Now, you know, in pharmacology, we learn the generic names, right? What are the brand names here? Viagra. Okay, and then there's another Me Too drug that was made by a different company. So sildenafil is the one that Pfizer has made all of their money. Pfizer used to be a small family-oriented drug company. And then they discovered that they had this phosphodiesterase for cyclic GMP. They were studying it in their cardiovascular division. They thought it would be kind of useful. But it turns out it really wasn't. So they said, well, we got this stuff. What are we going to do with it? And some brilliant Pfizer biologist said, oh, cyclic GMP regulates other functions. So they decided to take advantage of those other functions, and they made a big mint of money. Now they're Pfizer Global. They pretty much chewed up every other little small drug company in the world, and it's all based on Viagra money. So this has had a really big impact. And now we've got a lot of people that are using it for their erectile dysfunction. Then we have transducin. Now transducin is what's going to regulate this phosphodiesterase that's in the rod outer segment. So the transducin is the G protein that works with rhodopsin. And you can see that it's inhibited by this same Zapronast. So when you look at the side effects on the package insert of Viagra, you'll see that one of the problems that might occur is visual dysfunction. Because, you know, no drug is just a magic bullet for one little thing. So this makes sense now. If you've got something that works really well in regulating the smooth muscle vasodilation, it may have some other effects, and in this case, it's in the visual system. Then you've got some other things, some that are not sensitive to your methylxanthines, some that we don't really know very much about. So the whole point is that we can have some differences here that are based on what kind of phosphodiesterase you have in your cells. So these can be targeted so that you can use drugs that could inhibit one. And here we've got our PDE4 scaffolded on one of those ACAPs. It's regulated by PKA, but it can eliminate the cyclic AMP that might be in the vicinity of that PKA. So you can keep an area where you're not going to have a lot of cyclic AMP that's made, so that you can really target different regions even within the same cell. So here's our ACAP function again. This is called muscle ACAP, which is why it's got the little ACAP there. This reiterates visually for you to go back and look at how that catalytic subunit is going to activate the phosphodiesterase. Here are some other ACAP membrane preparations. You can see here that this PKA is regulating this subtype so that we can have our feedback mechanisms. Now, you can take a look at this slide at your leisure. This shows two different cells in an atherogenic lesion, and how we can modulate by using different kinds of phosphodiesterase inhibitors so that we can either target what might be in the atherogenic lesion plaque or the vascular endothelial cells. So depending on what kind of phosphodiesterase you have in your cells, you can use different inhibitors. Now let me close briefly, very quickly, to tell you about this rod outer segment, because this has rhodopsin, which is not found in any other cell except rods and cones. It's got transducin, which is a G protein that's not found in any other cells except rods and cones. And so when light hits, the retinal is already on this membrane in this little disc that's only found in rod outer segments. So it's light that's activating the retinal that's already embedded in the rhodopsin. So in contrast to all of those hormones that we talked about today that are going to diffuse in to activate their receptor, this already has the retinal there. It's this light that can activate the retinal. It's a lipid, it's got double bonds, it switches to an all-cis, it changes the conformation, and voila, it can activate this transducin. So now the transducin has the alpha subunit. Now this alpha subunit can interact with a phosphodiesterase, which was one of the phosphodiesterases on that list. Do you remember which one it was? Should we go back and look? Which one of these was the one that is in the rod outer segment? 6. Okay? So it's that unique phosphodiesterase, not found in other places. It's got alpha, beta, a couple of gamma subunits. It's got a delta subunit that I've been kind of very interested in, and anybody that's interested in finding out why I'm interested can talk to me about it later. But this now gets activated by the transducin alpha subunit. And when that happens, it dissociates these gamma subunits, and now these catalytic sites are going to be breaking down the cyclic GMP. Now, why would you want to do that? Because in those neurons, you have a cyclic GMP-gated ion channel that's going to be allowing calcium to come into the cell. And so when you can close that channel with cyclic GMP, it'll prevent the calcium coming in. But if you can break down the cyclic GMP so that it's not there, then you can allow the calcium to come in. So this whole signaling takes place very, very rapidly, simply because the only thing you have in these discs and these rod outer segments are all of these proteins that signal this response. So it's like flipping right through a whole series, just like you have for oxidative phosphorylation in a mitochondria. They're all compacted together and they all work. So we know a lot about this whole system, and the beta-adrenergic receptor wouldn't have really been well discovered if it weren't for what we know about this. So if you all have any questions, I'd be happy to